We're going to take a look at fermentation. Remember that fermentation is anaerobic, which means that no oxygen is used, so the electron transport chain is not going to be used. And in fermentation, only glycolysis is going to be performed. So remember that glycolysis only occurs in the cytoplasm, and that's true for both prokaryotes and eukaryotes. So fermentation is only going to take place in the cytoplasm. And the thing to remember about fermentation, besides not using oxygen and occurring in the cytoplasm and just using glycolysis, is that it's only going to produce 2 ATP per glucose. So you're not going to get very much ATP per glucose at all, so only 2. And the other important thing to remember is that the final electron acceptor, obviously it's not going to be oxygen because this is going to be used when oxygen is not available. So the final electron acceptor is going to be some sort of organic molecule that's going to be found inside the cell. Okay, and then this is just a nice diagram of fermentation, just showing you that we're going to take glucose, do glycolysis, break it in half, form 2-pyruvate. And remember, in glycolysis, the energy produced is just the 2 ATP. And remember, we're also going to be ripping electrons off glucose as we break it in half. And we put those electrons onto our electron carrier, NADH. And here's our little electron carrier, our little dump truck here, NADH. And there's our electrons in there. And usually, if oxygen's available, NADH will take those electrons and dump them off at the electron transport chain, and those electrons will go down the chain and then eventually get dumped onto oxygen. But oxygen is not available in fermentation, so NADH has to dump the electrons somewhere else. So where NADH is going to dump the electrons is onto an organic molecule inside the cell. So here's where we're going to be dumping the electrons onto this organic molecule inside the cell. And then our dump truck, our NADH is now empty. And then we can go back up and break another glucose down and make another 2 ATP, pick up some more electrons, and do another cycle of fermentation again. And then depending on what organic molecule the electrons get dumped onto, there's a lot of different end products that can be made in fermentation. So you can just look at this, you don't have to memorize any of it, but you can just see that this is where all of our products come from fermentation. So depending on what organic molecules the electrons get dumped onto during fermentation, we can make lots of different things because of fermentation. So that's pretty cool. Okay, but let's just take a look at 2 examples of fermentation that I know that you've learned about before. So here's lactic acid fermentation, which is what our cells do. So in our muscles, so maybe when you're sprinting or lifting weights, we can't get enough oxygen to our muscles, so we can't use our mitochondria and do aerobic respiration, so our muscle cells switch over and do fermentation. So here we are doing just glycolysis. We're going to break the glucose in half and make our 2-pyruvate. We've made our 2 ATP, and that's all we're going to make in fermentation per glucose. And then we've also ripped off electrons from the glucose as we break it in half, and we've put them onto our electron carrier, a little dump truck here, our NADH. And here it is, full of electrons. And if we had oxygen available, normally the NADH would go over to the mitochondria and dump those off with the electron transport chain, and then those electrons will go down the chain and end up on oxygen. But because there's no oxygen available, then NADH is like, well, I got to put the electrons somewhere. So here in lactic acid fermentation in our muscle cells, we're just going to dump the electrons right onto this organic molecule, pyruvate. And so we just dump them there, and that gets rid of the electrons, and we end up making lactic acid. So here's what we end up making in our muscle cells. And now our electron carrier, NAD+, is now empty. We can go back to the next glucose, be ready to accept more electrons as we break the next glucose apart and make another 2 ATP. Okay, and then just another type of fermentation just to be aware of would be alcohol fermentation, and this is done by yeast, which remember is a type of fungi. And this is going to be used to make alcohol and also used in making bread and other baking products. So this one, we're going to start again with glucose, doing glycolysis, splitting the glucose in half, making our 2-pyruvate. Along the way, we make our 2 ATP. We rip the electrons off of glucose and put it onto our electron carrier, NADH. Fill that one up. And then the organic molecule that we're going to dump the electrons onto, instead of dumping it onto pyruvate, the yeast just take one more step. They just take the pyruvate and they just chop off a carbon, and that carbon becomes carbon dioxide. And in baking, that's important because that's what's going to make the bread rise. So that's why you use yeast to make the bread rise. And then what's left is the organic molecule that we're going to dump the electrons onto. And then that is what makes the ethanol or the drinking alcohol. So that allows us to empty our dump truck here. And now our electron carrier is now empty, and we can go back to the next glucose and be ready to accept more electrons. Okay, and then here's just a nice summary of the 3 ways to break down glucose and make ATP. So you can just look at this and compare aerobic respiration and then anaerobic respiration and fermentation. Okay, but remember that there's other nutrients besides glucose. You can also break down triglycerides and proteins. So remember, triglycerides are fats, and it's a very common energy source for organisms. And here's a picture of a triglyceride here on the bottom. And you can see why they're called triglycerides. So tri meaning 3, they've got 3 fatty acid chains here in yellow, which are long hydrocarbon chains. And then they're going to be attached to a glycerol molecule here in brown. And glycerol is just a modified sugar there. Okay, so to break down a triglyceride, we're going to use enzymes called lipases, because triglyceride obviously is a lipid, so they named the enzyme a lipase. And so we're going to hydrolyze the triglyceride, and we're just going to break it apart into, we're just going to break it right here, right between the glycerol and the 3 fatty acid chains. So we're going to break it apart into glycerol and the 3 fatty acid chains. Okay, so the glycerol will just feed into glycolysis because that's a modified sugar. And then the fatty acids, what we're going to do with those is we're going to break those into smaller pieces. So we're just going to break them down into little 2-carbon pieces here. And so by doing that, we're going to convert the fatty acid chains into little 2-carbon pieces, and these 2-carbon pieces end up becoming acetyl-CoA. Okay, and then we can feed that into the Krebs cycle. So the fatty acids, when they're broken down into these little 2-carbon pieces, we call that beta oxidation. So that would be a good term to know. Okay, we can also break down proteins and make ATP from them as well. So on the bottom left, I've got a picture of a protein. And to break down the protein, we're going to use some enzymes called proteases. And to break the protein down into the individual amino acids, we're just going to break down the peptide bonds holding the amino acids together. And so that process is called proteolysis. Remember, lysis means to split or break apart. And then once we've got the individual amino acids, so like over here on the right, we've got an individual amino acids, then we're going to do what's called deamination. So D means to remove. So deamination just means that we're going to chop off the amino group and remove it from the amino acid. So deamination just means to remove the amino group from the amino acid. And then what happens to the amino group in us is we're going to convert it to urea. So our liver will convert that amino group into urea, which is a waste product, and that gets filtered out by our kidneys, and we excrete that in our urine. Okay, and then what's left of the amino acid, this little carbon skeleton, we can just feed into the aerobic respiration pathway and then make ATP from that as we break that down. Okay, and then lastly, this is just a nice summary slide that just shows all 3 different types of nutrients. We've got the carbohydrates here in the middle, and we can see going all the way down here, these are just all 4 steps of aerobic respiration. And then over here on the right, we've got our triglycerides, our fats, and you can break those down into the glycerol and the 3 fatty acid chains. The glycerol you can just feed right into glycolysis and make ATP from that. Remember, the fatty acid chains are really long, so you've got to break those into little 2-carbon pieces. That process is called beta oxidation. And remember, those turn into acetyl-CoA, and then you can feed that into the Krebs cycle and make ATP from that. And then we can also break proteins down and make ATP from that. So to break the proteins down in the individual amino acids, that's called proteolysis. And then once you have the individual amino acids, we need to chop off the amino group because we can't do anything with that. So removing the amino group is called deamination. And then we take the amino group and turn that into urea, which is a waste product, and we get rid of that. And then what's left of the carbon skeleton of the amino acid, we can just feed that in at various points of the aerobic respiration pathway and make ATP from that.